Morning. We will make a start. We're running a few moments late. I'd like to start by thanking you all for coming and welcome you to this combined seminar with Bevital and Olink covering multi-omics. We're delighted to have you here today to talk about this exciting opportunity of integrating both proteomics and metabolomics. Okay. For those that are joining via the hybrid, please let us know that you can hear everything. Post a question if not. Okay. So I will begin. First of all, I should say I'm Adrian McCann from Bevital. Sorry, I should have started with that. So all right. Okay, before we dive into the seminar, let me introduce you to Bevital. Okay. Sorry, I have a problem with the printer. Bevital is made up of a small team of experts, including laboratory technicians, data scientists, and researchers who work collaboratively to advance research in metabolomics. We specialize in targeted metabolomics, and we have a very strong track record of providing high precision, high quality metabolomics data for researchers investigating nutrition, inflammation, disease pathology, and physiology by targeting specific groups of metabolites and pathways. We provide scientific support from study planning to sample analysis to data interpretation, and for the last 20 years have taken great pride in our commitment to helping academic, clinical, and industry-based researchers apply targeted metabolomics to their chosen research field. And now, as of this year, we are also expanding to offer targeted proteomics as a service provider of Olink. Each of you have got a bag, those are in physical attendance, and within the bag you have a brochure or flyer with the Bevital offering included. You also will see an overview of the metabolites that we actually cover. You can look through this in your own time, of course. Moving along to today's seminar, as I say, this is all about multi-omics, also known as integrative omics. Multi-omics is a fascinating and relatively new approach to research that involves combining metabolomics, genomics, transcriptomics, and proteomics with the goal of gaining a more holistic and comprehensive understanding of biological systems by analysing various layers of biological information simultaneously. Today, we'll focus primarily on proteomics and metabolomics, and a quick overview of our agenda, as can be seen here. In a slight change, Carsten Nielsen from Olink will start and provide an introduction to Olink technology. He will be followed by Anoop Ambikan from Karolinska Institutet, who will present work investigating multi-omics in relation to severe COVID-19. Anoop will be followed by Olav from here at Haukeland University Hospital, who will present a study evaluating inflammatory responses within Graves' disease using both the Kinnurin pathway and an Olink panel. Lastly, we will finish with Sarantis, who will present on next generation proteomics in the era of omics and precision medicine. After the four speakers have presented, we'll open the floor to questions from the audience. That will be followed then by a very, very quick slide on your opportunity to win a free Olink panel and Bevital panel as part of a competition that we're running, okay? So, to begin, I invite Carsten.
Yes.
Okay.
Thank you very much. Does not work this one or?
No.
It'll work. Yes. My name is Carsten Nielsen. I work for Olink. I'm a field application scientist covering the Nordic territory. So if you have any question regarding the technology of Olink, you're more than welcome to reach out to me. But hopefully after this presentation, you'll know a little bit more what Olink is and what we can offer along with Bevital. So a little bit about the company itself. It's a Swedish company based in Uppsala, but we also have departments in the US and in Asia. We're working with more than 800 labs and institutions around the world. And we also work with the top 20 pharma companies in the world. They're all really interested in Olink technology because it has so many opportunities using this technology. And more than 190 labs around the world are using the Olink technology. We cover 100% of the biological pathways. I'll show you a little bit later how it looks in one of our programs, so you can actually visualize how the proteins are present and where they are. We have more than 1,400 peer-reviewed articles out there published with a lot of really great data. So there's always a lot that you can look at already, and more is coming week by week. Our technology is validated. I'll show you briefly how we validate all of our assays and how we can scale from working with only a few proteins to several thousand of proteins. So a little bit about Olink. We have a platform with our PEA technology, proximity extension assays, and we have a range of products ranging from a really high number of assays down to only a handful of proteins. So we have a technology that covers the whole scale, from really explorative to targeted approach when looking at proteins. And we have the tools to actually visualize the data as well. So a little bit about the PEA technology. How does it actually work? The proximity extension assay, it works. We have a dual binding. So we have two antibodies that need to recognize a protein. On these two antibodies, we have a nucleotide, a single-stranded DNA attached. So on both ends, when they're closely in proximity, they will ligate to each other, and in combination, they'll produce this double-stranded DNA. So this is actually how we translate a signal, a protein signal, into a DNA signal. And then we can use the technology developed for DNA to actually amplify the signal and get really good results. And that is why the technology is so sensitive and so specific as well, because we need three things to happen. The two antibody needs to bind, and that we need to have the matching DNA as well. Otherwise, you will not get a signal. So that is why the Olink technology is very specific. And we can measure this in very small volumes and in really high multiplexing. And this is a little bit about our portfolio, our platform. We have different offerings. And last month, we sent the newest product on market, Explore HT, where you can actually look at up to 5,300 proteins as in a little as 2 microliters of sample. So this is really extraordinary. We also have the Explore, where we have the 3,000 different proteins, where you can run them in panels of around 384 different assays in each of the panels. These technologies are read out on NGS systems because there's so much data, so much assay on there. So it needs the Illumina system. But from the Olink target, it's a targeted approach where we use the qPCR technology. And that is what the Bevital have today in their lab. And that's where they can help you with the target platform. The target platform runs in the 96-well format, as you might already be familiar with qPCR. So it's quite easy to handle. And the combination of all of these panels, you have around 1,100 proteins you can detect with the target technology. We also, as you can see here, the readout is as NPX, normalized protein expression. That is what we're using in Olink. We should use it for comparing groups. So it's not, it's all relative. So you can measure absolute with some of our other products, but what is interesting is to look at differences between groups. So in theory, it doesn't matter if you have a readout in picogram per milliliter or you have it in NPX if you're comparing. The targeted is a cytokine panel, only uses 1 microliter, and we have a flex where we have a library of 200 assays that you can pick and choose and make your own panel, selecting from 14 to 21 different biomarkers. And Olink Focus is something that we can produce customized to include any proteins in the whole library, and we can optimize it for different sample types as well. These technologies are validated and tested for plasma and serum, But as I will show you, we have a lot of other matrices that it will also work with. A little bit about what historically has changed with the technology. Before, it was low sensitivity, lack of specificity. You didn't really know which protein you are picking up. You need a lot of sample to do this, and you have a limited dynamic range. But all these things we have overcome with our PEA technology. So we have high sensitivity. You can measure really, really low concentration. We have exceptional specificity. We have the three things that needs to happen before we actually measure, so we know exactly which protein we are measuring. You need a minimal of sample volume, as less as 2 microliters to get 5,300 assays. That is quite unique. And we have a large dynamical range, and not only the dynamic range, but also the biological relevance range. That is the most important thing. And how does it actually work? This is the qPCR readout. I'm showing you here the target platform and what is run here in Bevital. We have a qPCR reaction where we have the two antibodies binding and then the nucleotide binding, as I told you. And then we have an amplification step. And then we have amplification and detection using microfluidic chips. If we are working with a really high throughput, the first two steps will be the same. Then we'll put library adapters on and then sequence it on the Illumina platform. But what is unique also that for every step in the protocol, we have actually controls in here as well. So when you're running a 96-well plate, for example, we have all these controls in each individual well alongside your samples. So we know exactly what is going on with the assays at every step of these steps here. And in addition to that, we also have a row with different controls in. So we are very sure that we know exactly what is going on when we're using the Olink technology. So it's built-in quality. And as I said, it's tested and validated for human plasma and serum, but we can also run with a lot of different matrices. I think the only two that we have not good results is feces samples and FFPE. That gives a little bit struggle with the technology. But everything else that we have tested works with the Olink technology. There might be some sample preparation and something you need to think about, buffers and so on, but all of those things we can help you with. So please reach out if you have some really interesting matrices and want to run, we can set something up. And as I said, you only need 1 microliters of samples going into the assays. And why does other researchers actually use the technology or scientists? Well, it depends on where you are. If you're in an academic institution, you're interested to understand biology as, or if you're running in a pharma company, you want to patient stratification. If you already know by taking a blood sample, if this patient group will actually be beneficial of getting this drug, it's a huge benefit that you can know that on day 1 and not after 1 month or 2 months of treatment. So if you already have that data, it could be really, really powerful to stratify patients, for example. But a lot of different ways that you can use the Olink technology. And a little bit about our validation. We're really proud of the work that actually have gone into develop this technology and these assays. So each individual assay of these more than 5,000 that we actually have gone through this validation. It's inspired by FDA. We are testing the specificity, so we know that it's only picking up this protein and not something else. The sensitivity, we do a dilution curve to know exactly what is the limit of detection of this particular assay. And also the precision we're testing from one run to another, but also from one lab to another lab. What do we actually expect of precision when using this technology? And the same with scalability. Does it work as great if you run in single plex, only look at one assay? Does it perform the same if you're looking at 5,300? And that we have all the data tested and validated, and also the dynamic range. And we make sure that it's biological relevant as well. So it's what you can actually measure in a human body. And it's very transparent. So this is an example from one assay, we have it available. So if you have an interest on one specific assay, for example, just reach out, or you can actually look it up on our homepage, and you can see the data we have for each individual assay. And just to get an idea of the work that has actually gone into producing this 5,300 high quality assays, is that we in Olink, or our R&D department, they have screened more than 15,000 protein assays, but they need to pass this very strict QC criteria, otherwise it will not end up in our products. So it's really important for us that what you get out and the results you get out at the end is something that you can actually trust. And the last thing I want to cover is our online tool. It's called Olink Insight. There's also the link here. It's a free tool you can use. You just need to sign up with your email address. You have access to a lot of really interesting things on Olink Insight. For example, we have a pathway browser based on the Reactome database. So for example, you have a list of 13, 50 proteins that you're really interested in your kind of research. You can just upload the list in Excel CSV, and you can actually, then it will be mapped out in this pathway. You can actually see visually how where these proteins are in the pathway. So it's really a nice tool to work with. You can also use it for panel selection. If you have your favorite 30 proteins you want to analyze, you can copy paste them into the program or upload a list, and then it will tell you need to run this oncology panel, because then you hit 28 out of the 30 proteins that you're interested in. And you can also search publications in there, what has been done, is in the literature with this particular protein of interest that you have uploaded. So there's really a lot of opportunities. We also have normal ranges. So if you're looking at big cohorts, what do we normally see in a healthy population? We also have data in there for COVID-19 story. We also have disease atlas regarding cancer. So a lot of research has gone into actually how you can look at different cancers, compare one cancer type to another, and see what proteins are actually different from cancer type to cancer type. And all these data are available and intuitive to work with. You can stratify, looking at different age groups, BMI, regarding the normal ranges, for example. And we have just included, or recently included the UK Biobank data in there for the normal data. If you're not aware, we have run in Olink more than, I think it's 55,000 samples for the UK Biobank with the Olink technology. The first half of the data is released. It was run with the Explorer with around 3,000 assays, but only half of them are available now. But the later half will come. Is it later this year or maybe next year? But it will be available at some point. And we also have data from the China Kandori Biobank as well. So really, a lot of great data is already available to look and have a play with. And to put into what we started with today, it's the whole approach. What we actually want in Olink is enable the understanding of the real-time human biology. And as a part of this, proteomics play a really big role, but it can, if you want the complete understanding of human biology, then it will be really interesting to have the metabolomics, transcriptomics, and genomics as well. So Olink will play a big part in getting the proteomics part so that we can really understand what is going on in the human body. And that was it for me. And I'll be here the next couple of hours after this is done. So if you have any questions, please feel free to come up and ask them to me, and I'll be able to help you. Thank you. Yeah. You want to use this one?
Okay, good morning, everyone. So my name is Anoop. I'm from Karolinska Institute, Stockholm. So today I'll be giving a talk on how we analyzed different kind of omics data in a COVID related study. So I'm currently working as a bioinformatician in a system biology group at the Karolinska Institute, Stockholm. So in this group, so we mainly study host metabolic reprogramming in related to different infectious diseases. And we also, our line of research is to metabolically engineer to identify potential host-directed antiviral strategies to all the viral infections. Then we hypothesize to repair immune cells through metabolic modulations. So usually we start the study without a hypothesis, and we will analyze the study data first, and we will look for where we can make any hypothesis out of the data. Then we will try to prove the hypothesis in the lab. So that's how we do our research in the group. So in today we'll talk about more merely on SARS-CoV-2 and how we can integrate or how we can analyze different kind of data, and if we have a lot of data. So I will focus more on the technical part because I'm an informatician. So I just want to introduce some few concepts using which you can actually derive more meaningful information from the multi-omics data. So you know that when we get an infection, the virus hijack our metabolic system, and they do in response to that, the body will have a lot of metabolic reprogramming to fight the virus. So that is already known. So what we can do to identify all these metabolic reprogramming. So that's what I will show. So I will introduce a term. I don't know if any of you heard of the term systems biology. So our perspective is to look at the system in a system wide view. For example, if you look at the figure, so if you concentrate only one part, you may not know what is happening in the other part. So this is our main idea. So we have to always look at the system from the outside in the wide perspective. So that is actually what the system biology is doing. So system biology actually try to find the interaction of different components of the system and try to tell you what is happening in the system. So in the first study, what we did is we started the study in vitro. So we did the study on Q7 cell line, and we generated proteomics and transcriptomics data from the cell line. So then after that, we did create a network analysis or using proteomics and transcriptomics, and I will show you how we did that one. So in this figure, it's actually there's no pointer. Yes. This one. Yeah. Okay. So this is the network we created using transcriptomics data. So in this, maybe difficult to see. Each dot is represent each gene. And here each dot is represent each protein, and the association between the gene is measured based on a Spearman correlation. So what we did here, we created the network, and we look at the topology of network, and we try to find is there any communities of co-expressed genes exist there. So we hypothesize that if you find any communities like this, that may perform a coordinated function in the disease. So we did create the network in using transcriptomics and in a proteomics using data. Then we identified is there any communities of genes or protein exist there. Then when we integrated using functional association. So here in this you can see each one is represent each community that we shown here, and this barbell is represent these communities here. Then we look at is there any community share any common function. Each link in this figure is represent the common function. Then we've identified that some of the communities in proteomics are functionally associated with the transcriptomics data. So that community is very specific to COVID, meaning because this we identified in both the transcriptomics and proteomics data, which is more robust result. Then we look at what are the function of these communities. Then we identified those proteins or genes which belong to mainly in HIF1 and TNF mTOR signaling pathways. So this is our first study which we did in vitro. So then we collected a sample from the patients. For the hospital, and we mainly studied 4 different cohort. So we have healthy controls and COVID infected generally, and the COVID infected we classified them into 2 groups, like a COVID severe and COVID mild. So the definition of mild and severe is based on oxygen consumption, and it was done by the clinicians when they arrive in the clinic. So we generated we took the sample from them, and we generated metabolomics and transcriptomics data, and also we also have Olink and single cell type metabolomics data in this study. So first we analyzed the Olink proteomics data and the metabolomics data. So here in this column is all the comparative analysis between each pairs, like healthy versus COVID. And since our focus is more on the severe disease, so the last column is represent the analysis between hospitalized, severe, and mild. And we found quite a few chemokines are significantly different at COVID severe compared to mild. And we also analyzed the metabolic stat on the same cohort. And we look for is there any severity specific biomarker is there based on the metabolic stat, and we found that mannose is identified as one of the biomarker of a COVID severe disease. So then, so the question we are thinking is that all this characterization of the cohort is purely based on the oxygen requirement and also the decision by the clinician. So we were thinking like, is this the right way of classification of the patient? Because this study was designed in the first wave of the COVID before the WHO registration was came. So we were thinking, why can't we use the metabolomics and the transcriptomics data? So can we group the patients based on the data alone? So we used a technique called similarity network fusion to merge the metabolomics and transcriptomics data to cluster the patients. So this is how the result will come. So this is actually the sample to sample similarity using a transcriptomics data, and this is the using metabolomics data. So what does the method do is it will calculate the similarity between each sample based on the data, and it will make a network of transcriptomics and metabolomics data. Then this network they will do the overlap. Then the method will try to find a common like a similarity of samples from the using the both network. Then it will remove the it will only select the common link, and it will show us the cluster of existing cohort. So we using this method, we actually found 4 patient clusters, and we named this cluster 1, 2, 3. And you can see that in cluster number 4 is mostly all the healthies came here, and this darker green is actually health COVID negative and antibody negative, and this is actually the lighter green is a COVID negative, but we have identified antibodies of COVID, so we classify the antibody positive COVID negative. So and in cluster number 1, so this darker orange is a severe. So we saw that one of the cluster is mostly COVID mild based on a clinician's perspective, but 2 of the severe is came to cluster 1. So that we believe that maybe this 2 patient is actually wrong classification from the clinician perspective, or maybe they are overreactive to the COVID disease, because one of the doctor they claim that some of the patient is very scared of the disease, so that makes them difficult to breathe. So because of that, maybe they end up in the ICU. So maybe this is a wrong classification. But in actual scenario, based on that data, is found to be a mild COVID patient. Then we also have identified some of the mild patients clustered together with severe. So maybe they are acting mild, but based on the data sets, that is their molecular signature is more similar to severe. Then we look at their interleukin levels based on the Olink proteomics. Then we see that is in cluster 3, which is this one, is high. So this is and the level of interleukin is cluster 1, which is this one, is slightly lower than severe. So this actually makes us much more robustness of the clustering. So we believe that this clustering is worked perfectly. So all the downstream analysis we used this classification rather than the clinician's patient classification. So here, since we have transcriptomics and proteomics data, so here we did we did create a network of transcriptomics and metabolomics data together. So here in each link represent a significant association between either gene to gene or gene to metabolomics or metabolomics to metabolite. So and when we did the topology analysis, we identified 8 communities of strongly related metabolites and genes. And then we look at the expression of their expression at a severe compared to mild to find is there any community specific for the severe disease. Then the red color here represent the fold change of expression at severe compared to mild, and we can see that in C1 is mostly upregulated in severe compared to mild. So we can clearly see that this cluster is more specific to the severe condition. Then we look at what are the function of this metabolite. So before that, we also see that this mannose, which is found to be a biomarker of severe disease, also falls into this cluster, so which will also give us some more confidence to say that this cluster is something specific to COVID severe. Then when we look at the function of this cluster, we mainly identified this is mostly belong to HIF1, FOXO, and RIG-I signaling pathways. Then we did some mathematics-based analysis. It's called a genome scale metabolic modeling. So here the model, which means that a mathematical representation of all the metabolic system in a tissue. So this model is nothing but a representation of each reaction in a metabolic pathway in a series of mathematical equation. For example, in the form of A plus B gives C plus D, where A and B be a product, reactant, and C and D is the product. And with the enzyme, which is either catalyzing this reaction, or in case of a transport reaction, that the gene which is transporting this metabolic in and out of a cellular component. So we have a reference metabolic network is already available, which has the model of all the reaction curated so far in a human tissue. So we took that model, then we integrated the transcriptomics data of our from our sample, which we generated from the COVID infected patient, and we integrated that information in the model. And the final output will be a model which is specific to the COVID, meaning that model will contain only the reaction which is active or in a person when get a COVID. So once you have that model, we can do a mathematical procedure to find what are the metabolic reactions specifically which is happening in a COVID body. So because that is very important, because when we analyze simply the transcriptomics data, if you analyze any transcriptomics data generated from any infectious disease, it's always a metabolic pathways come into most significant. You will find T cell cycle, you will find amino acid synthesis. But for example, if you analyze HIV and COVID or any other infectious disease, if you find a T cell cycle at the top, then can we say that the 3 viruses use the same mechanisms? Or inside the pathway, there are any difference is that. So using this method, we can actually identify inside the T cell cycle which particular reaction is different in these 3 viruses. Maybe the reaction, maybe the difference is at the malate to format converter or in any other part of the T cell cycle, because T cell cycle is a series of reactions. So in the series, we could actually pinpoint which reaction is actually changing in a person gets in a disease. So this is the overview of the method which I explained right now. So the process of identifying the reaction which is changing using the genome scale model is called the fresh balance analysis. So I will explain. So when we have the model which is specific to the disease, then we have to define an objective function. And after defining an objective function, we will use a mathematical procedure. So it will tell you the all contribution of the rest of the metabolic reaction for the maximum production of this objective function. So here the objective function we used is ATP hydrolysis. So we hypothesize that if a cell has a more ATP hydrolysis, means that the cell is in a good growth condition. So we define the ATP hydrolysis as an objective function, and using that objective function, we calculate what are the contribution of rest of the metabolic reaction for the more production of the more activation of the ATP hydrolysis. So we did the same thing in using our COVID data. So we identified that few of the reaction which is specifically active in COVID severe cases. When we look at deep, we found that those reactions which are active in COVID is mainly a transport reaction, which is transporting TCA cycle intermediates from cytoplasm to mitochondria, mainly transporting succinate from cytoplasm to mitochondria, transporting alpha ketoglutarate from cytoplasm to mitochondria, and even same for succinate and fumarate. So this tells us that in our analysis tells that when a person gets COVID, there is a more activation of this lower part of the T-cell cycle in COVID. So we did actually validate the result by checking single cell type transcriptomics data. How we did is that, so since we identified this reaction is active in COVID, so we checked that in which, since the data is from a whole blood sample, so there can be different kind of cell types. So first we identified in which cell type this genes is more expressing. Since we don't have the single cell type data, so we took a publicly available single cell transcriptomics data which has similar design. Then we look at this particular transport genes are expressed in which cell types. So we found that these genes are mainly expressed in monocytes. Then we isolated the monocyte from our samples. Then we did metabolomics on those monocytes cell types together, and we identified that all the concentration of alpha ketoglutarate and fumarate and malate is high in the monocyte, which is actually validated our findings using our computational prediction. So the outcome, these are the main outcomes of the my multi-omics analysis. So we identified the signaling pathways such as HIF1, mTOR, and TNF signaling is highly associated with the SARS-CoV-2 infection. Then we also identified that mannose and increased glucose level is a biomarker of the disease. Then using the genome scale modeling, we identified transporter genes which are transporting CCS cycle intermediates, which has a crucial role in COVID disease in severe case. So that is from me. So I'd like to acknowledge my team member, and thank you so much for the Olink to invite me for the presentation. Okay.
So Olav. What?
Yes, hello everyone. I'm Olav Ueland from ophthalmologic department here at Haukeland University Hospital, and we work in close relationship to the endocrine department here. And thank you for the opportunity to present our experience in using proteomics and metabolomics for exploring inflammation in patients with Graves' disease, with and without orbitopathy. First, I will give you a brief introduction to the subject, and then I will talk about our first study where we explored inflammation in Graves' disease using proteomics by Olink, and then our second study where we used metabolomics from Bevital in the same group of patients. And finally, I will show you use of Proteomics in another endocrine disorder and conclude. Graves' disease is one of the most common autoimmune diseases. And TRAB is essential. Most often it's stimulating, leading to hypothyroidism in these patients. And 4 out of 5 patients are women. And these might experience symptoms of activation shown in this picture. And among others, they can have eye symptoms. 40% of the patients have more or less eye symptoms. And this is an autoimmune inflammation in the orbital tissue affecting the extraocular muscles and the orbital fat. And clinically, there's a difference between the active inflammatory phase and the inactive phase. On the upper picture, there's a patient with active orbital inflammation where there's swelling and redness periorbitally in the eyelid and in the conjunctiva. These patients often experience pain and pressure especially when they are moving their eye. This swelling redness disappears gradually, and the patients go into the inactive phase where there's residual findings with which could be upper eyelid retraction, exophthalmos, meaning that the eye is pressed forward, or strabismus. This significantly interfere with the patient's quality of life. We have a helpful tool called the clinical activity score, where 1 point for every symptom or clinical finding is given. And if a score out of 3 out of 10 or more, then clinical active inflammation is present. There are several risk factors for development of thyroid eye disease in patients with Graves' disease. And the most important is smoking. Smokers has an increased risk of 7 times increased risk of developing thyroid eye disease compared to no smokers. And there's been extensive research in different tissue in blood, tears, and urine to find a biomarker for thyroid eye disease. But the only biomarker in clinical use is TRAB. This is a specific and a sensitive biomarker for Graves' disease in general, but not for thyroid eye disease. All patients with thyroid eye disease have positive TRAB, but not all patients with positive TRAB develop orbitopathy. Our first study addressed to identify a biochemical biomarker predicting risk of development of thyroid eye disease or presence of thyroid eye disease in patients with Graves'. And this study was based on the National Norwegian Registry of Organ-Specific Diseases called ROAS. And in this register, 100 patients with Graves' disease were included between 2013 and 2020. And the register included clinical data and serum samples. And we used 120 healthy controls for comparison. From these 100 patients, 36 had thyroid eye disease, 29 at inclusion, and 7 developed eye manifestations later in this disease course. Out of these 29, 7 had active inflammation with a CAS score above 3 or more. And we used this proximity extensive assay technology from Olink, which was nicely presented in the previous presentation, where DNA marked labeled antibodies bind to different inflammation-related proteins, and we used this panel with 92 different inflammation-related proteins. And And out of when we compared healthy controls to patients with Graves' disease, we found a significant difference in 52 out of these 92 inflammation-related proteins. And this is a volcano plot where we can see that these 10 biomarkers were decreased and these markers were increased in Graves' patients compared to healthy controls. And the most increased biomarker was TNF receptor superfamily number 9. And as a biomarker for Graves' disease, if we performed a ROC analysis where we found area under the curve of 80%. But this is inferior to TRAP, which is already in use. But we looked specifically after a biomarker for thyroid eye disease. So for the subgroup of patients with thyroid eye disease compared to patients without thyroid eye disease, we found that it was significantly increased levels of macrophage colony stimulating factor 1 and interleukin 6. And interleukin 6 is known to have a central role in the orbital inflammation, but here we show that it was also increased systemically in serum. And here we can see the serum level of interleukin 6 in healthy controls in gray, in Graves' patients without thyroid eye disease in blue, and patients with thyroid eye disease in red. And we can see there is an overlap between the levels, and therefore the interleukin 6 is not useful as a biomarker for Graves' disease. But then we compared the patients with serious TED to patients with less serious TED, and we found that a protein called FMS-related tyrosine kinase 3 ligand was significantly increased in patients with severe disease, eye disease. This is a protein responsible for mobilizing of hemopoietic stem cells, which could be relevant in the pathogenesis of the disease, because we know that recruitment of immature fibroblasts to the orbital tissue is essential in this condition. But we wanted to find a biomarker for pointing out patients which is susceptible to develop thyroid eye disease, which are prone to develop thyroid eye disease. And we found fibroblast growth factor 21 to be significantly elevated in patients without thyroid eye disease at inclusion, but who later developed the eye disease in the disease course. And we can see that the fibroblast growth factor 21 was increased significantly both between the Graves' disease without thyroid eye disease and the healthy controls. So this could be a potential biomarker for thyroid eye disease, but it has to be explored further in future studies. We found interferon gamma and interleukin 6 both to be significantly raised in patients with Graves' disease compared to healthy controls. And both these two inflammatory biomarkers are inducers of tryptophan degradation to the kynurenine pathway. And following this line, we performed this study, but the purpose was to characterize interferon gamma-mediated inflammation and metabolites of the kynurenine pathway in patients with Graves' disease. And in this study, we analyzed 34 biomarkers in the same group of patients that was used in the previous study. It included 10 metabolites of the guanine pathway and 3 indexes. And in this study, we used 100 match controls for comparison. And here we can see we compared healthy controls to patients with Graves' disease. And we found it was a significantly raise in the red dots are raised biomarkers and the blue are suppressed biomarkers between Graves' patients and healthy controls. And we found increased level with strong effect size in 7 biomarkers in Graves' patients, where the 6 most increased were all from the Karolinska pathway. We performed the same analysis between patients with Graves' disease and without, but we did not find any differences between with and without orbitopathy. And we also performed a factor analysis, which explained 40% of the variation between the biomarkers. And this 2-factor solution showed that the factor loading with Th1 and cellular immunity biomarkers loaded was most strongly associated with Graves' disease, but also a factor consisting of acute phase markers were also associated, but to a less degree to Graves' disease. So this shows that there's Th1 and cellular immunity present systemically in patients with Graves' disease. Our data is to some degree reproduced. This is a study from Annelien Stockland, which is in our group, and she performed the same proteomic analysis in another group of patients. And also in this group of patients, the most elevated biomarker were TNF receptors to become the number 9. And in this study, follow-up study, the patients were treated with antithyroid drugs for 12 months. And we can see that the total load of inflammatory markers is decreasing during the 12 months treatment. And when we look specifically at the TNF receptor superfamily number 1, we can see that it's gradually decreasing during the treatment period, and it's approaching healthy controls after 12 months. And after 12 months, most of the patients are cured. And finally, just for comparison, this is a study in another endocrine disorder with slightly cortisol overproduction. And when we look at this volcanic plot, we can see that there's a different alteration in the biomarker levels. In this condition, there is more of cardiovascular profile, and the cardiovascular inflammatory biomarkers is dominating compared to patients in Graves' disease. So in conclusion, using Olink and Bevital, we have demonstrated a distinct immunologic profile. You can see that there's a immunologic fingerprint of patients with Graves' disease. And combining Proteomics and Metabolomics, we got a deeper insight in the mechanism in the meta-analysis, showing that there is a Th1 immunity present in these patients. So, yeah, thank you all for listening, and thank you to Mark.
Thank you very much, Olink, for putting this meeting together. Today, I will try to show you some examples how proteomics can enable multi-omics studies. together with metabolomics, of course, and why it's important to use an omics approach, especially when you are dealing with complicated disease. I will start this slide with explaining our portfolio in Olink. We try to cover all the aspects of the drug discovery process, for early drug discovery, preclinical research, up to phase 1, phase 2, and phase 3. We have examples and collaborations with pharma companies and academic institutions, and a lot of papers, and please feel free to reach out to us later on to discuss and get more material about your project and your research area. We are really proud because we are the only company provides NGS proteomics. And this, I will show you later on why it's important, the NGS proteomics. Later, our latest actually baby, and we're really proud of that, is the Olink Explore HT. It's a platform that now provides scientists with a number of more than 5,300 proteins based on NGS approach. We have an increased library, a simplified workflow, higher throughput, and automatic data analysis. We are a company that is continuously innovating. We have started June 20 with the first NGS platform having 1,500 proteins. In June of 2021, we had double the proteins, there are 3,000, and now we are like more than 5,300 proteins. And here's why it's important to have a high throughput. You can analyze, as I mentioned, more than 5,000 proteins. We use a minimum input of serum or plasma and other matrices. We can go down to 2 microliters with a dynamic range of femtograms to milligrams, and then it down to NGS. Coming back now to why it's important the NGS, why it's important to have so much data, right? And why you have to have an omics approach. We hear nice talks from my colleagues before about multi-omics in a different disease area. Now we are talking about complicated disease. The modern disease that are based on lifestyle changes, for example, right? They're complicated, and we need a lot of data, and we need omics integration in order to understand disease and create precision treatments. Here I try to depict most of the most important, let's say, disease areas where we have generated a lot of impact, like Olink, and where we have created a big amount of publications. I can start from neurodegenerative diseases, cancer, but also aging-related diseases, BMI, metabolic syndrome, so on and so forth. The scope of my presentation is to show you some examples and how people they have used the proteomics in conjunction with metabolomics and other omics data to analyze and understand this complicated disease. Let me start first with a little bit of introduction in the field of proteogenomics. A very important factor is the pQTL. pQTL is a SNP. That this SNP is regulating the expression of the gene. If this SNP is close to a gene in a 1 megabase, we call about cis-pQTL. If it is far away, it's called a trans-pQTL. And they're really important because this pQTL, in a proteogenomics approach, they're potential targets for drug discovery, but also for biomarker discovery. As an Olink, we are part of a lot of consortia. Our first baby and our first consortium, we're really proud, is the Scallop one. The Scallop that is run by Anders Malestig. Now we are more than 40 investigators, more than 43 cohorts. And the idea is to understand in a populational study, disease and precision treatment. The first paper came a couple of years ago. We analyze cardiovascular proteins connected to cardiovascular disease in more than 30,000 individuals. And we were able to identify more than 400 new pQTLs. As you can imagine, this is a really powerful tool to understand and identify potential novel biomarkers. And here some list was validated already biomarkers, new targets, and reposition of already existing ones. Another great example, a little bit more recent, earlier this year from Claudia Langenberg's lab, an amazing work in proteogenomics field, where the group tried to understand how they can use proteins in conjunction with genomics data, NGS approach, in order to understand biomarkers for metabolic disease. They use our platform of 3,000 in more than 1,000 patients. And here's the main findings. You will see that they were able to, of course, report previously existing PQTLs, but also identify a big number. You will see in your right hand with the orange color, novel candidate of causal genes. And here I summarize the main finding of the paper. More than 250 previously unreported PQTLs were identified. And it was a first time that we'll be able to address the correlation and the causality between 224 cis-PQTLs and 575 health outcomes. Again, how important is the multi-omics approach? How important is to have a big data set in order to understand complicated disease? We are really proud because we are proud of the biggest, to my knowledge, epidemiological cohort study in the world. Is the UK Biobank. There are more than 500,000 participants. We are teaming up with the biggest pharma in the world. The first round of the data came out earlier this year and includes proteogenomics data of more than 53,000 participants. And the findings are really striking. We're able to assess for first time in such a big study correlation between protein expression and different covariants. And as you can imagine, it opens a really great world in the early discovery and not only, let's say, research area where you can follow, for example, changes in BMI by following expression or differential expression of proteins. And I can tell you that there are a lot of projects and papers based now on this data, and you're going to see a lot of things coming in the upcoming years. Stay tuned, or you can reach out to us to get to know a little bit more, because I think it's a big initiation and a lot of data that they can dig and get a lot of information out of that. In the field of metabolic disorders, but this can be applied in different complicated disease, you see that this is somehow the workflow, right? You can see the circadian rhythm. Let me see, put a pointer here. You see circadian rhythm, obesity can influence and can be connected to metabolic disorders. Stress, I can put also smoke. Now, companies like Bevital and Olink help us to understand the multi-omics perspective, help us understand the phenotypes. And this is by screening plasma or serum, but also other tissues, in order to understand in a multi-omics perspective, how this complicated disease, how these complicated actually pathologies are expressing and are giving the phenotypes, because it's a some of them are multifactorial, right? They are connected to cardiac dysfunction, they are connected to metabolic dysfunction, and it's good to have the real phenotypes in a non-invasive way, checking plasma and serum, to understand exactly the pathology. And contemporarily, there are studies that are running in parallel or before or after even, where with different models, organoids, cell cultures, people, they try to understand molecular mechanism. And here's actually the workflow. And here in the multi-omics perspective, where companies like Bevital, as I mentioned before, and Olink help us to understand the multi-omics perspective. Some examples. I pick up this example because it's one of the really great paper where we try to understand for the first time biomarkers connected to heart failure based on right ventricular dysfunction in human pulmonary arterial hypertension. It's a disease, it's a malfunction of the heart, and for the first time, using a biomarker multi-omics based approach, combining mass spec data and transcriptomics from tissues, from heart, Olink proteomics in plasma, and you can see here from the Venn diagram, we are able to narrow down the number of biomarkers from hundreds potential candidates to a handful of them. And there are 5 that are connected to this heart failure condition. And even checking a little bit closer of these 5, we were able to see that LTB2 has the highest AUC among the others. A great example where you can start using multi-omics in order to narrow down the targets, narrow down the potential biomarkers that helps, of course, in the prognosis and the treatment of a disease. How multi-omic signatures now can help to understand challenges and changes in BMI. And of course, how this can be used to monitor changes in lifestyle intervention. A great paper this year from Watanabe and al., where they first try to see in all omics aspects, which of the omics approach are more easy or are correlating better, predicted and measured BMI. And as you can appreciate, on the x-axis, they are predicted BMI, y-axis is a measured BMI. They try different omics approach, also clinical laboratories and standard measures. And you can appreciate that metabolomics and proteomics, they are highly correlating predicted BMI and measured BMI compared to any other omics data. That's really a nice example for the omics approach, and a nice example of the value of proteomics and metabolomics. And going further, using machine learning algorithm, they were able to see that metabolomics data and proteomics data, they're able to predict changes in BMI in high accuracy, compared even to clinical and standard measures. The combined omics is equally or a little bit higher than the proteomics and metabolomics itself. I mean, you can see really clearly that the value of metabolomics and proteomics and the value of combined omics in understanding BMI changes, and probably later on using like a proxy for changes in lifestyle intervention. Now, I wanted to bring this example here because I like the holistic view of this paper, where they try to see changes in the equilibrium, especially reflected in the macrobiota homeostasis, how it's connected to inflammatory bowel disease risk, and how the combination of multi-omics approach, how the combination of proteomics, metabolomics, and genomics can help people to understand mechanisms. It can help people to understand how a disease can progress, and of course, target later on therapies more efficiently. Here is actually the description of the project. They are working in science. They work in a wellness program for more than 2,800 participants. They perform a multi-omic phenotype using metabolites, proteins, but also clinical assessments and microbiome. And together they perform a DNA sequencing. They were able to see that there are variants in the gene DUOX2. This is a gene that is responsible for hydrogen peroxide. That means that this gene is helping to kill the bacterial, actually the pathogen bacterial in gut microbiome. And they were able to see that there was some rare mutation, loss of function of this gene in this population. Now, combining the genomics data, the mutations, with the phenotypes that they have recovered mainly with metabolites and proteins, they were able to see this really nice scheme. Again, the participants, the mutations with the DNA, and as you can appreciate for this, different mutations, rare mutations in this population, it was associated with IL-17C levels, high levels. What does it mean? If they went back to the mouse model, again, the nice framework, how you can combine different type of approaches and preclinical, early clinical studies, you will see that mutants on this gene in mice, they don't attack, they don't produce hydrogen peroxide. We have invasion of the pathogens and the epithelial cell. We have stimulation on inflammation pathways. IL-17C is increased. Now, the nice thing, going back to the human population, they were able to see that these mutations that they have screened before are connected to IBD. And this is a nice example how you can use the omics, how you can use different models in order to see mechanisms of action, how to see mechanisms of progression of a disease, model the disease, and develop targeted therapy. This is from this slide. I would like you to pay attention only in the scalability, Not because the other parts are not important, but I'm sure that my colleagues before they give you a nice introduction about that, but because we are really proud to have the first LDT together with Octave. This is a company in US, Octave Bioscience, the first LDT for assessment of multiple sclerosis. As you may know, multiple sclerosis is a very expensive and difficult disease, has really poor outcomes and difficult prognosis. We team up with the Octave Bioscience, where we use our flexibility capabilities. That means we first start with a discovery approach in more than 300 samples. We validate then this discovery in a different cohort of 600 samples, and we identify 21 proteins that are behaving exactly the same way by testing them, and all this by testing them in another independent cohort of 600 samples. And we are able to see that 18 proteins, they behave in a way that can predict with a high precision the progression of multiple sclerosis. Of course, Octave there has integrated other data there, like clinical measures and other omics data. But it's great to see that we have helped them to develop the first LDT that is close to be approved. I think it's already approved for multiple sclerosis. A nice example how you can use the same exact technology, starting from the discovery approach to more targeted validation and having a panel dedicated to your disease for early prognosis and diagnosis. I will stop here just to show you that we are a company that is expanding not only innovation, but also we providing great science. We are nowadays more than 1,500 publications, and we are raising. And I would really encourage you to visit our website. We have a nice database called Olink site where we are starting introducing population studies and disease. You will find the great paper from Matthias Ueland, the great data from the Matthias Ueland cancer disease atlas. And there you can see potential biomarkers for different cancer types based on proteomics, but also distribution of proteins in healthy populations and in COVID-infected disease patients. I'm sorry I spoke too long, and I'm really happy to take your questions and your comments. Thank you very much.
Okay, thank you, Sarantis, and thank you to all the speakers. I think you'll agree they give a fantastic overview, not only of the technology underlying Olink, but also the potential of combining omics approaches, in particular metabolomics and proteomics, and as Sarantis pointed out also, the microbiome now more recently, the newest of the omics children as such. As Sarantis says, and the other speakers, we're open the floor now to questions. Please feel free to ask. Maybe you could begin by indicating who your question is for. That would be helpful. Yes.
Yes, no, I have a general question, yes, about the availability of the data. I mean, there is now apparently a lot of data coming out, and so I'm just wondering, when you publish these data, what are the requirements to make them publicly available? I mean, if I do an experiment that is almost identical to somebody else, can I mine their data, or do the authors select themselves which data they're going to make available?
I can attempt an answer to that, and maybe Sarantis can also join in.
Yes.
As far as I understand, it's very much dependent on the publication that you're targeting at the moment. I don't think there's a standardization or consensus in terms of what data should be made available. Ultimately, I guess my personal opinion is that when it comes to sharing data, I think it's in the interest of everyone that we do that. I think from Olink's perspective as well, I think they have a very transparent approach to the data sharing once it's published. Sarantis, could you add to that maybe?
Yeah, I will add to this. Thank you very much. I think we don't have a policy, let's say, for the data, and the only thing that the data, they don't belong to us, right? Scientists, they come to us and do the data and belong to scientists. That means we have to agree, or we have to put you in contact with the scientists, and then you can decide with the scientists that have generated the data what they can share, how they can share, what they can, because it's not an Olink proprietary, the data. That's the thing. We don't own data. On the other hand, we have our database, the Olink Insight, where you can access and see what we can share with you. But this is because we have already a consensus with the scientists to upload their data there. We are in process of developing. We are in process of taking feedback. We don't have any policy in place, but the thing I can say, we are open in science, we are open in discussions, we are open in exchange of data, of course, having the consensus and on board the scientists that have generated and created them. That's in a nutshell.
Thank you, Sarantis.
You're welcome. You're welcome. I can see also some in the chat box, right? Hans,
Okay.
Olav, can I answer them or you want me to do it?
Can you do it here?
Yeah, so I can see the question. So
Okay.
Yeah.
Sorry. So we have a question for Hans Olav. Hans Olav, why choose the use of serum instead of plasma? Thank you in advance.
It is for me. Can I?
Available.
Yeah.
Yes, we need be based on the who was registered where serum samples is then.
Yeah.
For every patient, we didn't have it. Yeah.
Just to follow up on that, typically when it comes to biobank samples, often you'll collect and process your biological material, sometimes years in advance of knowing what you're going to use that material for. So it's often a case of you using what material you have. If you have serum, then that's often the only one that you'll be able to use. If you have serum and, say, EDTA plasma, you then have the choice to make. At least from a metabolomics standpoint, the matrix can impact some of the analysis. We have a lot of experience when it comes to metabolomics. From a proteomics standpoint, I think you guys also see differences in terms of the protein profile, whether you use serum or EDTA plasma, for example. So I think in that instance, it's always best to talk to the people either at Bevital or at Olink directly and get their input and advice on which is the best matrix to use for the metabolite or the protein that you're particularly interested in.
yeah, and the project as well.
And the project, of course, as well. Thank you. Yeah.
And maybe you can just repeat the answer for Hans Olav's. It was just he had serum available, right?
Yes.
Because that person won't hear from.
Sorry.
Yeah.
Okay. Okay. Yes, Sarantis, you.
Yeah, just also to mention, we do have a preference as well. I mean, depends. I mean, we can work with any of the matrices and with high efficiency. We have data to support things that are connected to stability, for example, and storage. Yeah, depends the project, depends the scientific question that you have to raise. But for us, it's we can work with any matrix that you can imagine, and even more.
Thank you. Yusa.
Yeah, thank you very much. Very interesting. You said that also mice serum can be analyzed, but what about rats?
Straight to you, Sarantis. Rat samples, I assume, is not a problem either.
No, it's not. We have a mouse panel for that. We have a dedicated new mouse panel dedicated on cytokines, but we have dedicated products for mice. Yes, and for sure we know that the NGS platform can be used for non-human primates as well with a high efficiency.
At least we have some data from rats.
I'm sorry, I cannot hear you.
Yeah, we have some data for rat as well. So
Yeah.
The flexibility itself is a little bit lower when you share different one animal to another, but we have data. So we don't detect all the proteins, but at least you can get some.
like it's over there. So you answer the question.
Next question. Okay, I'll take another question from the team. Sorry. Question for Bevital and Olink. Can you analyze metabolites from soil, air, microbiota, and foods to capture a planetary health perspective of one human status or disease? I will begin, at least from a Bevital perspective. We do not analyze metabolites in soil, air, or microbiota. We typically measure samples in serum or plasma. We can also measure cerebral spinal fluid, CSF, urine. We're also capable of measuring cell lysates, cell lines, and lysed tissue samples. We have the capability of measuring microbiome-derived metabolites in circulation, but not any of the others. And from an Olink perspective, I think that's the same. It's typically biological
exactly
material.
the same. Exactly the same. Our antibodies is targeted mainly human proteins. I mean, not mainly exclusively human proteins. That means we cannot see metabolite microbiome proteins, but we can see the effects that microbiome has on humans, right? That's what we are looking.
You can take one from the front.
Yeah, I do that.
Yeah, I have I guess a technical question for Olink. The PEA, I guess technology, if it's two antibodies, does that mean it's two separate monoclonal antibodies that, bind to different epitopes?
Carsten?
Yeah.
So are you also,
Yeah.
so are you developing, are you actively developing your own antibodies?
Yes, we have a company, Agrisera, that only owns and provide us with these antibodies. So that's the key ingredients in Olink technology. And that's also why we have purchased and developed and working with them, because we want to have the technology internally, because it's a high value in our products, in our antibodies. And so it's a key part. Yes, so we are producing it ourselves.
Okay. Kenneth.
Yeah, thanks. Thank you for all the great talks. So I have some experience in mass-based proteomics. So why should I migrate from this mass-based proteomics to the Olink antibody-based proteomics? I didn't catch that. Maybe I missed it. But...
Yeah. Did you get this question, Sarantis?
Yeah, I think it's like why I should change mass spec and take proteomics, right? That they take Olink proteomics. That's the question, right?
Yes.
Okay, I like this question. Thank you. You don't change. We don't want you to change, and you should not change. There are two different technologies, and you take different outcomes. If you go and you know better than me in plasma serum analysis with mass spec, definitely you don't capture low abundant or low molecular weight proteins. And we know this because we are working with mass spec guys like Stephanie Hauk, Matthias Mann, and we get a really feedback from that we add value actually, because we can capture low abundant proteins, we can capture smaller molecular weight proteins that mass spec easily cannot capture. The other thing that I can say is we can go really high throughput. We can work with 2 microliters and can go really high throughput. That mass spec for the moment is not something that it competes. For this, I don't see like a competition. We don't see competition with mass spec. We see a complementarity. And mass spec facilities now worldwide, they really acknowledge this and include us to their pipelines. But I can give you some data also if you want for different matrices like dry blood spots, for example, how we can work together with mass spec to obtain optimal results. I hope that I answer your question.
Yeah, thank you. Got a thumbs up. Okay. I will take another question from the teams here. Again, based question directed at both Bevital and Olink. In case we identify new key metabolites for health from the Gates Foundation big study on the food periodic table of elements, aimed at identifying 400 new bioactive compounds from 40 major food types, will your catalogue of biomarkers expand to include those new ones? Might be new nutrients of importance, or nutrition would also be important. I think as a summary of this question, Sarantis, the individual is asking, will you add proteins or will we add metabolites to reflect the observations that they have and maybe a more untargeted or exploratory approach initially? I will answer for Bevital first, maybe. Yes, from our perspective, we are very open to adding new biomarkers of interest. What we always try to do is have biomarkers that are closely related or complementary to what we already offer. From a nutrition standpoint, that has been like the core of what we've done for 20 years with our measurements of B vitamins and fat-soluble vitamins. So that type of project would be well aligned with our approach in terms of expanding to include biomarkers that are related to nutritional status. Sarantis?
Yeah, I mean, I can tell for the moment, I can tell that we are working with the Gates Foundation US. We have some projects that we're running together already. Unfortunately, I cannot share more at this stage, but yeah, we are open to develop all the time. We have also a special link in our webpage, if I'm not mistaken, Carsten, correct me if I'm wrong, where we accept, let's say, suggestions for hot biomarkers, because we are really expanding on this. But yeah, Carsten, please feel free to add also your perspective on this as well.
Yes, we want to include as many as possible. Now we have 1,300, but of course we want to include everything that you can find in the body at some point. And we have a part of our homepage. If you have requests for certain proteins that we do not cover, you can actually put them in, and our R&D team will look at these proteins and see if we can include them in new release of products going forward. So this is the way you can sort of try to push if there's something proteins that is of interest for your research.
Thank you. Greta first.
Yeah, we have performed several Olink analysis in hormone, and that was unfortunately before you made this normal range, which I think is a major improvement. Do you have any idea about the day-to-day variation, circadian variation, the impact of food, and aging status on all these markers? Because that's coming when we are trying to publish the studies.
I don't know if Sarantis have any ideas.
I haven't. I'm sorry, the sound goes really bad, and I could not hear the question.
So,
if you don't mind to repeat it, yes, please.
Yeah, I just summarized the question. So one of the audience members has asked if you have an overview or understanding of how different factors might impact the protein profiles that you observe, such as the impact of circadian rhythm,
Amazing,
fasting status, day-to-day variation, for example.
Amazing question. Thank you very much for giving me this opportunity to answer this. I can tell you from my experience and discussions that we have with people like Steve Horvath with the circadian, works a lot nowadays with the clocks and circadian clocks and proteomics clocks and aging clocks. Definitely proteins are more stable and less sensitive to changes than transcriptomics, for example. And when Steve went to do DNA methylation profiling and correlate with expression or with the proteins, he sees more nice correlation with proteins than with expression. For sure, it's a biology behind, and for sure, not all the proteins they not behave the same. Any condition is different, but definitely proteins are more stable and less sensitive to changes than the transcriptome itself, and more close to what's happening in nucleus, actually. For this, it's a nice proxy for phenotyping screenings rather than the transcriptomics itself.
Yeah. And just to follow up from a metabolomics standpoint, we know that there are certain metabolites that are impacted by fasting status, for example. Increasingly, we know that circadian rhythm and other external or environmental factors can impact the metabolite profile. From a research or scientific standpoint, what we would encourage is that people are aware of these factors as much as possible, and that you adjust either statistically in your modeling or at least in terms of your interpretation when it comes to concluding what your data suggests. That was what I would add. Just.
Yeah.
I'm going to take a question now from the teams again. This one is for Anoop. To have this multi-omics integration, how big is the cohort? Or is there a minimal participant number that is required to overcome biological variation bias? Yeah, you want to come up and speak?
Yes, okay.
Question to the.
Yeah, if you come here to the.
There is a very difficult question because there is always the two aspects. So one is aspect of a statistics and one is aspect of a clinical side. So it's always good to have a good number of subjects to have a maximum statistical power, but sometimes it's very difficult to recruit that many patients. So there's nothing we can do about it, but it's always good to have a more number of subjects.
Thank you. Thank you. One last question from Per Magnan.
Just follow up your question. Yeah, CMS, I totally agree. It's not a competitor, a complementary. And that leads us into the area of isomers. Most proteins have 2 to 7 isomers. In some occasions, 1 or 2 of these are pathogenic, the other neutral. So that leads us also to comparison between the Olink assay and your competitor, the Suma, the aptamer base. As you probably know, the Olink has lower precision and more hits. And that probably reflects that these 2 assays, even when targeting the same protein, doesn't capture the same isomers because of different specificity. How do you approach this problem in the future?
Yeah. Did you get the follow-up question?
Yeah,
Sarantis?
if I got the question now, it's like the question between Soma and Olink, right? And then how you discriminate the 2 technologies and what one technology captures compared to the other, right? That's pretty much the the nutshell.
That was the initial comment, but the follow-up question was, how does Olink technology aim to overcome challenges with the different isomers for individual proteins?
Carsten, you want to start, or do you want to?
No, you can go ahead. Yeah.
Okay, super. No, because I thought that you were wanting to say. I thought I heard your voice for this. I stopped. I'm sorry. No, I mean it's clear. Thanks. Great question. There are two different technologies. We see two different things. One thing is based on aptamers and sees something that could be a protein but could not be a protein. We know that the rate of unspecificity is quite high, and something that also SOMA agrees and acknowledges this. The other technology is based on polyclonal antibodies that capture proteins. Now, of course, different isomers of proteins could have an effect, but I think at this point, SOMA were targeting specific conformation of the protein rather than the protein conformation itself is more sensitive than us. Now, how you can design now antibodies of specific isomer, that's a big challenge and a big discussion. Is the same topic of how we can use, let's say, for different modifications like antibodies. We are looking on this, we are checking on this, we are open in development and revolution. But at this point, yeah, I would trust more if you want to go like for post-translational modification or for a different isomer, I would trust more the mass spec rather than the SOMA at this point, right? But coming again to SOMA and Olink comparison is what these two technologies they are capturing. So it's an aptamer capture a conformation of a protein or could be something like a protein, and an antibody target protein. And so usually a polyclonal antibody, that means could capture different conformation change of the protein itself. I don't know if this answers your question, but I'm happy also to answer with any more comments that you have on this and discuss more if you want.
Yeah. I think we can always follow up after the fact if any questions are pressing, or if anyone didn't get a chance to ask something that they hope to. In the interest of time, we have to push on a little bit, okay? I won't keep you much longer, folks. I would just like to move on now to
maybe. Yeah.
Okay, this slide. I mentioned at the beginning, there's an opportunity to win a free Olink panel and free Metabolomics panel at Bevital. This will be a pilot study for the analysis of 88 plasma or serum samples across Olink target 96 or 48 panel and a Bevital platform of your choice. To have the chance of winning this, and this is both for the physical members here as well as those who are joining us online. To have the chance of winning, you're required to upload a 250-word abstract to the page here by the 31st of October, describing the research question, detailing the background, your objectives, the pilot study design, and indicate your chosen Olink panel and Bevital Metabolomics platform. For more information, you can follow the link here. This gives you more information on what the competition entails in terms of what we require from the abstract, as well as other information in terms of terms and conditions. Okay. I think, yeah, and we will also circulate this link afterwards to everyone in attendance, both physically and in the hybrid form. Okay. Folks, that is everything from us today. Thank you, Sarantis, again. I'd like to thank all of the speakers in particular for joining us today, both physically and online. And thank you also for joining us, the audience here, again, both physically and online. Please feel free to follow up with any of us, either at Bevital or at Olink after the fact. After the fact, we really welcome any questions that might come up from this seminar, and always welcome the opportunity to speak with fellow scientists and researchers. For those that are physically here, could I ask you just to sign the attendance if you haven't done so already before you leave? Okay, thank you very much, folks. Have a good day.